My name is Sarah McMullen, and today I will be doing my Exam 3-1 video describing a prokaryotic replication fork. A prokaryotic replication fork is formed in response to DNA strands being separated at the origin of replication to allow for transcription to occur. The origin of replication is a specific nucleotide sequence where replication begins. Helicase, this dark blue enzyme here, is an enzyme that unwinds the DNA. It's done by breaking the hydrogen bonds between the nitrogenous base pairs. The replication fork, just right here, is when, as the DNA is unwound, it creates the Y-shaped structure, also known as a replication fork. You can see if you trace this structure, it creates a Y shape. As the replication proceeds, single-stranded binding proteins, these little purple star things, coat the single strands of DNA near the replication fork. This prevents the single-stranded DNA from winding back into a double helix, thus allowing the replication process to continue. DNA polymerase III is an important enzyme that adds nucleotides one at a time to the growing DNA chain. DNA polymerase III is only able to add nucleotides in the 5' to 3' direction, since a new DNA strand can only be extended in this direction. So that's why we have a lagging strand DNA polymerase III and a leading strand DNA polymerase III. They are working in opposite directions of each other. In order for a new nucleotide to be added, it's required that there is a free 3' OH group. The next nucleotide will be added by forming a phosphodiester bond between the 3' OH end and the 5' phosphate end to the next nucleotide. RNA primase, which is this orange-brown here, synthesizes an RNA primer that provides a free 3' OH end to start the replication process. Now, DNA is unwound in the replication process into two strands. The strand that is continuously synthesized towards the replication fork and is complementary to the 3' to 5' parental DNA strand is known as the leading strand. That is this top one up here. The lagging strand is the other strand that is complementary to the 5' to 3' parental DNA strand. The lagging strand extends away from the replication fork and is composed of small fragments known as Okazaki fragments, these little red bits. The direction of the leading strand will be 5' to 3', while the direction of the lagging strand will be 3' to 5'. Okazaki fragments make up the lagging strand and are a building block for DNA synthesis. Each fragment requires a new primer to start the synthesis process. Gyrase, which is this donut shape up here, is an enzyme that is a type of topoisomerase. Gyrase helps to prevent the overwinding of the DNA double helix ahead of the replication fork, as the DNA is opening up. It's done by causing temporary nicks in the DNA helix before it reseals them. DNA polymerase I replaces the RNA primers as synthesis continues. So as you can see down here, we have DNA polymerase I, but there are no more primers coming down this way. The DNA polymerase I then breaks down the RNA and fills in the gaps with the DNA nucleotides. The process leaves little nicks between the newly synthesized DNA and the previously synthesized DNA. This is where ligase comes in. The nicks are sealed by ligase, which is an enzyme. Ligase catalyzes the formation of the phosphodiester linkage between the 3' OH end of one nucleotide and the 5' phosphate end of the other fragment. This allows DNA to be formed in the 5' to 3' direction. I hope you enjoyed this video and found it helpful.